Hello, this is Dr. Dan.J.Guerra. I come to you from Authentic Biochemistry Studios in the Inland Pacific Northwest. Today is remarkably the 21st of January, 2022. That's correct. So, as I promised on the podcast feed, what I plan on doing is giving you a general lecture on membrane biochemistry, with sort of an emphasis on the understanding of how the lipid composition regulates the fluidity of the membrane, and therefore at least one element of its function. Okay, so let's just do that, shall we? Okay, so we'll do the slideshow, and we'll start from the beginning. I hope that you enjoy this, by the way, because I enjoy, whenever I can, talking about lipids. So again, general biochemistry lecture. Authentic Biochemistry is what you're listening to and watching. This is me. My name is Dan.J.Guerra, and I am your host and professor. So let's get started. All right. So, whoops. You saw it all. Okay. What do membranes do? They serve as semi-permeable isolating barriers to separate contents of the cell, whatever that happens to be, whatever kind of cell, from the external environment, or pathway constituents within organelles to coordinate overall cellular homeostasis and even organismal homeostasis. There are proteins in cell membranes, and they have lots of functions. And typically, these are what are emphasized when you get a membrane lecture in biochemistry. However, that's not what I emphasize, except if I'm particularly interested in some membrane protein. I would like to really emphasize the macromolecular structure of the membrane. And of that, you need a deep understanding of the lipids that make up that. At least first level compositional ordering of it. And then eventually I will talk about interdigitation, particularly of the various classes of lipids and how they make up the unique membranes that you see, right? But proteins are involved in transport of substances across the membrane. Of course, there are enzymes within the membrane, both the plasma membrane and all the endogenous membranes, such as those that encompass the organelles associated with various functions. So peroxisomes, endoplasmic reticulum, the nucleus, mitochondria, of course, endosomal compartments. These are all membranous subcellular structures. Some of them have more proteins than others, but they all have a unique repertoire of lipids and indeed proteins. Okay. So going back to the list of proteins, receptors, particularly the plasma membrane, that's very important. On the surface of the cell, you have a host of receptors. And I know that all of you are fully aware of this because if you're interested in biochemistry, you're going to know a lot about this, right? What kind of receptors? Well, we'll talk about those in specific, but you have them on the exterior surface, and those tend to bind ligands, for example, a hormone or a growth factor. And then you have also many proteins that are embedded within the lamella, that is the inner and outer leaflet of the membrane. Then you have proteins that extend throughout the membrane, cap out into extracellular space, extracellular matrix, for example, as well as enter deep into the cytosol. And then you have some proteins that are only on the internal leaflet of, let's say, a bilayer membrane. Okay, so they all have different functions. They are organized and distributed within that lipid matrix during their synthesis, coming usually from the ER, the endoplasmic reticulum, through the Golgi apparatus, and then ultimately fusing with, as a lipid raft component, the growing plasma membrane. Now, there are other proteins and other lipids which play a major role in what ends up in that ER Golgi translocation. In fact, some of them bypass the ER altogether, and even the Golgi to some extent. So there are remanufacturing of lipids, particularly a very long-chain polyunsaturated fatty acid containing phosphoglycerol lipids and sphingolipids from the peroxisome, but also the mitochondrion has a great deal to do with the manufacture of the biosynthesis and distribution of the lipids, which make up that component of membrane once translocated to the plasma membrane. Yeah. All right, so I want you to understand that. Also, the nucleus plays a role because sometimes we get microRNAs that end up moving through the plasma membrane. Right? We talked about those a great deal in various lectures. I won't go into it right now. The other thing you have are mediators on membranes, another protein. These protein mediators aid ligand receptor complexes by triggering a sequence of events. Typically, this would be something like G protein coupled receptors, or let's say calcium-mediated acetyl phosphate generation from endogenous plasma membrane phosphatidylineacetyl bisphosphates or trisphosphates, and then generating after phospholipase activities of various kinds, phospholipase A2, phospholipase C, phospholipase D, you're going to generate a myriad of secondary messengers, which will then translocate information from the externals of the cell to the internal regions of the cell. And that could include all the way from promoting transcription and therefore involved in chromatin retailoring, or maybe epigenetic modification so that there are specific genes that are expressed first transcriptionally and then translationally and then post-translationally, all based on the signaling that was received by the plasma membrane, by those kinds of proteins that are embedded within that membrane. Okay. Right. Denylate cyclase is another important enzyme that you find associated with the plasma membrane, right? And I know we talked about that a great deal, so I want to get you to have your bearings of where we are. So again, what does a membrane do? It has selective permeability. So that means it will allow some substances to cross and some not. This has a little bit to do with partition coefficients. We'll talk about that later. That means how lipid soluble versus aqueous soluble a given molecule might be. But that's not the only thing, because there's also facilitated diffusion, right? And energy transduction to pull, for example, nutrients from the external milieu to the internal regions of the cell for purposes of, say, oxidation, for ultimately purposes of bioenergetics that generate ATP. Okay. So you have channels and pumps. Those are primarily used for ions. Those can include sodium, potassium, calcium, chloride. Those are 4 favorite ones, but there are others, right? In fact, there are also charged organic compounds. And those would include what we're calling substrates here. Then you have specific receptors for signaling, and some of those can be hormone receptors. Think insulin receptor, glucagon receptor. Various hormones such as alpha melanocortin stimulating hormone, those kind of proteins, peptides, which will bind to a receptor on the surface and then carry out some signal transduction, which ultimately lead to some sequelae in the cytoplasm, or maybe again, significantly nascent transcription based on a signal that came through that receptor-mediated process. And you also get an exchange of materials from the extracellular environment. So some great deal of, for example, glycoproteins leave the cell and end up having most of their function extracellular. Think about immunoglobulins. Think about cytokines, chemokines, growth factors, matrix metalloproteases, things we've talked about in the past many times. All of those have to make it out of the cell, right? And so there are processes in and out of the cell we have described in various lectures, and we'll do some throughout these membrane lectures. And those processes are exocytosis and endocytosis. So exocytosis means sending something out in bulk package or bringing something in bulk package. We talked about endosomes, and we talked about porosomes, and we talked about clathrin-coated pits, various kinds of translocation phenomena. We talked about synaptosomes, translocation phenomena that have very discrete lipid components and therefore lipid specific geometric structures, which allow for those kinds of bulk processing of moving, transporting molecules that are favored for a given signaling or an expression of an intention of the cell to administer some kind of surveillance or indeed even a direct immune response. We talked a lot about that when we got into the T cells and B cells and of course dendritic cells and macrophages and etc. Now I put this one slide in here to remind everybody that membranes age just like the cell does, just like people do. So sarcopenia is a skeletal muscle contraction and chronic myopathy. Cardiovascular disease, that's where the cardiac muscle contraction is associated with heart disease, and that is sometimes associated with atherosclerosis, hypertrophy, myocardial infarction, and stroke. We've covered these in earlier lectures, and we will a little bit as we proceed. There are peripheral cell membrane disruptions, and those receptors and the channel-mediated injuries that are triggered by that response will then induce the production of reactive oxygen species, and either subsequent to that or adjacent and parallel to that particular eruption of reactive oxygen, you can also have phospholipase A2 activity, which could often release very long-chain polyunsaturated fatty acids of either the omega-6 or the omega-3 positional and geometrical isomers of fatty acids. And then those can be functional only after the production of eicosanoids or oxygenated fatty acids. And those enzymes that carry out the introduction of molecular oxygen, those preformed very long-chain fatty acids, include cyclooxygenase and lipoxygenase, among others. Right? We've talked about immune cells. We just mentioned it again. We know that immune cells are highly communicative across that peripheral cell membrane. We talked about extracellular matrix proteases often, then signaling for autophagy and apoptosis for cellular fate. And then, of course, you can also get necrotosis and ferroptosis, right? We talked about the immune sequelae associated with those unusual programmed cell deaths. The neuronal membrane corruption is usually or often, when it's in the central nervous system, linked to microglial-induced inflammatory responses. And this can be quite detrimental, but also it is necessary in the CNS to take care of any potential infection or inflammation, even if it's induced by other microglia, but certainly if it's coming from the periphery. Okay. So we talked about senescence a lot when we did the aging lectures. We talked about astrogliosis. Again, that is an inflammation of the astrocytes. We talked about MS, AD, PD, PFD. These are all diseases of the central nervous system, right? So multiple sclerosis has to do with the removal of the myelin sheath, corrupting nerve transmission. There's a plethora of information that comes out of Alzheimer's disease, Parkinson's disease, and prefrontal dementia. We talked about some of those cellular pathologies. Many of those are associated with membrane leakage or membrane corruption of some kind or another. So these are linked as chronological and stress aging phenomena. We also can talk about toxins, including certain pharmaceuticals at a certain concentration or chronic utilization. We talk about hemodynamic agents. We've talked about ethanol and encephalitis after pathogenic infection. We talked about the blood-brain barrier integrity loss. Finally, I can mention to you about membrane integrity linking long-distance innate and acquired immune cell cytokine chemokine growth factor stimulated cellular necrosis and or oncogenesis. Again, other forms of cell fate. Okay. So you have membranes for specific compartments. You've got organelles with specific function. And I've already mentioned to you that they're going to have unique membranes, right? So mitochondria and plasma reticulum, Golgi apparatus. One of the things that membranes are doing are specifying and localizing specific enzymatic pathways. Also, of course, when you're dealing with any reactive membrane surface, you're going to have excitation and response to that of a coupling mechanism for transducing that signal. And these can be chemical, neurochemical, and neuroelectrical, depending on what kind of reception is involved, what kind of membrane is involved. And of course, you also have with energy transduction, the mitochondrial membrane. Both membranes are involved with that, right? We talked about electrotransport chain. We talked about proton pumping across the inner mitochondrial membrane. We talked about the proton motive force. We talked about the ATPase. In plants, you've also got a chloroplast, which is also double membraned. And the galactosyl diacylglycerols that associate with the chloroplast membranes don't just end there with the chloroplast envelope. You also have the granule stacking and the thylakoid membranous system within the chloroplast. These generate surface area and allow for light reactions during that phase of photosynthesis, driving photophosphorylation, for example, ultimately the production of NADP, and then all that linked to the uptake of carbon dioxide after, of course, catalase reactions. And that will ultimately lead to the utilization of atmospheric CO2 to produce the disaccharide sucrose in plants, right? So there's a lot of business going on there in the membranes that we have touched upon in multiple lectures. I'm just reminding you here. Of course, you also get, again, oxidative phosphorylation, which is absolutely key to bioenergetics. Absolutely central are these membranes. So internal water also has to become compartmentalized. So if you think about intracellular fluid is about 2/3 of the total water, and it has high concentration of potassium ions and magnesium. Phosphate is also a major anion. Protein concentration is higher intracellularly, so there's an osmotic gradient involved. And then you've got the extracellular fluid. That's about 1/3 of the total water in a given organism, particularly thinking about an animal. Extracellular fluid is high in sodium and calcium. And the major anion extracellularly is, of course, chloride, Cl-. And typically glucose concentrations are higher extracellular rather than intracellular. Okay. So you can see here from this little chart I put on this slide, various concentrations of the extracellular fluid versus the intracellular fluid for sodium. Extracellular much higher, there's 140 millimole per liter versus only 10 intracellular. Move on down here to bicarbonate. You've got 27 millimolar extracellular, about only 10 millimolar intracellular. Phosphate, 2 outside the cell, a full 60 millimole per liter internal. And so this membrane then will generate a concentration gradient across it. And because of that, you're going to be able to drive ions in one direction or another. And with that transducing, you're going to change the polarity of membranes, okay, in terms of charge. And that change in polarity will then yield new, now modified, and then becoming reified signaling mechanisms and translocation of nutrients and waste products back and forth, etc. Okay. So this now allows us to start talking about the membrane itself. Of course, the membrane composition is going to vary with function, and that means with different cell lineages. So the major lipids in mammalian membranes are phospholipids, glycosphingolipids, and cholesterol. By phospholipids, I mean phosphoglycerol-based lipids. Okay? And the sphingolipids, of course, have a sphingosine base. And then the other very important membrane lipid in animal cells, particularly at the plasma membrane, where it's most increased in molar concentration, is cholesterol. Okay. So with a fatty acid, remember you have a long hydrophobic alkane chain, right? And then you have a hydrophilic so-called polar head group. That's not always the case, all right? But in the case of this would be a lipid. Now, in the case of a fatty acid, you're going to have a carbonyl or a carboxylic acid, and then you're going to have the long aliphatic chain ending in a methyl group. There are double bonds in there, of course. You're going to add a little bit more, because it's more oxidized, ability for that molecule to interact with aqueous dissolved substances. Right? But if you have a complete alkane, that fatty acid is going to be internalized in the membrane. And because of the hydrophobic effect, that membrane will be highly enriched as a hydrophobic center, right? A three-dimensional matrix. And it's going to be reified there by cholesterol, which adds some rigidity to the membrane and therefore allows for specified water conducting channels, which will bring things like ions across and glucose, etc. Okay. So there's this long hydrocarbon chain, for example. Sorry, let me pull myself over there. This is just a bunch of methylene groups. If there's no double bonds, it's not interrupted. And then you have that methyl group terminus. So there's the carboxylic acid. It's always oxidized, right? pKa of that in physiological state, you're never going to have this as a carboxylic acid with the proton. Okay. Here's oleic acid. That's one very important monounsaturated fatty acid that you do find in membranes. And you can see it as a double bond right in the middle of the molecule. So it's a delta 9, 18:1 fatty acid. 18 carbons at the delta 9 position. The 9 position of the carbon, there's 18 of them there. So it's right in the middle, and that's a cis configuration because the two protons are above the plane of the double bond. They're not trans, where one of these protons would be down and the other would be up. And you do have trans fatty acids in many, many, many membrane lipids. Okay. So when you get a double bond, it puts a kink in the molecule, shown here. Right? And so you get this so-called rigid bend in the hydrocarbon chain, and that then results in a new morphology of the membrane. And that change in morphology or shape will, of course, modify its structure totally, and then its function. So the double bonds in fatty acids play multiple roles, and one of them is to coordinate the macromolecular geometry and spatial isomery of the membrane itself. Okay? And that will lead to a whole host of biochemical and physiological functions, event ontologies. Okay? So here again are some of your standard fatty acids: stearic acid, oleic acid, linoleic acid, and alpha-linolenic acid. So you can see the number of double bonds varies with these. All these are 18 carbon fatty acids. Stearate is a complete alkane chain, or no double bonds, so you just have methylene groups. And again, the methyl terminus, there's your carboxylic acid shown protonated here. Oleic acid with that single double bond at the 9-10 position there. Linoleic acid is going to have a delta 9-12, and because 12 is 6 carbons away from the terminus, that would be an omega-6 fatty acid. And there's alpha linolenic acid, and that has a delta 9-12-15 double bond configuration. There are 3 double bonds in alpha linolenic acid. And because that last double bond from the methyl terminus, that is at the carbon 15, we call this an omega-3 fatty acid. It's omega-3, it's omega-6, it's omega-9. This is simply 18:1. Okay. So now we have a discussion of soaps and detergents. So very simple soap is something like sodium dodecyl sulfate. It's going to have a negative charge on one end, a strong positive charge because of the sodium atom on the other end, which is going to be associated with this molecule. Then you're going to have this aliphatic region. So because you have an amphipathic molecule, it means charged on one side or other with 2 opposing charges in terms of valency. You also have this hydrophobic region. That's why it's a soap, because soap can then dissociate membranes. It's the primary thing that soaps can do. Triton X100, which is a common detergent that's used in isolating membranes and purifying proteins in the biochemistry laboratory, is nothing more than a polyethylene glycol derivative. And it's where the polyethylene glycol is one of the terminal hydroxyl groups has been converted to a corresponding 2,4,4-trimethylpentanyl 3-il, phenyl ether. That's right out here. Yeah. And so it actually acts as a non-ionic surfactant, again, disrupting membranes. And that's what Triton X100 is used for. When you're isolating proteins from cells, you can throw in Triton X100, and that will disrupt the membrane structure because of its amphipathic nature. And because of that, then you can isolate proteins that might have already been embedded in the membrane or closely associated with it. I'm just showing you a little bit of, these are non-biological lipids, right? That's what those are. All right. So now you've got a lipid here. It's the glycerol lipid. There's the glycerol backbone, carbon 1, 2, 3. Okay. And so you have here this hydrophilic head group. And I know because it is a trimethylated ethanolamine. Okay, that's what this is. That's going to be choline. That's choline. When you have the phosphate ester, that's phosphonocholine right there. That's what that structure is. And for PC, phosphatidylcholine, that is called the polar head group. Okay. Then you've got, that's on the 3rd carbon. Then you've got carbon number 2, which had an hydroxyl, because after all, this starts off as glycerol. Right? So you have an oxygen ester now with that carbonyl, both at the 2 and the 1 position. This particular lipid, as you can see, is a 1 steroyl, 2 oleoyl, 3 phosphatidylcholine. That's its actual structure, right? And so this is a particular lipid you can find in the membrane. And notice that you have now different molecular species that can erupt. Depending on what fatty acid is in the 1 or 2 position, this lipid will have a completely different chemical and therefore physiological character. Okay. So these long chain fatty acids, even when they have double bonds in them, like oleic acid only has 1, this is still going to be a highly hydrophobic region. And so understand that this membrane lipid, that polar head group, is going to make it as a surface region of a bilayer, which is going to be more in contact directly with the aqueous milieu. Then internal to that bilayer is going to be all of this hydrophobic region, or aliphatic hydrocarbon, right? And that's how you generate such structures as bilayers. They form spontaneously because of the chemistry of water and the hydrophobic effect, which we've talked about in great deal. And don't worry, I will discuss it again, probably, if not in this lecture, in a subsequent lecture. We talk more about the biophysics of this whole system and how it relates to the biochemistry. Okay. All right. So here are some of the so-called hydrophilic head groups of major glycerol phospholipids. And you also find these unsphingolipids. So it's not just the glycerol lipids that you get these, okay? So take a look at them with the glasses back on. It's kind of hard to read that with that white background. Again, here is your preformed glycerol lipid, fatty acid in one position and on the two position. See them as oxygen esters with the carbonyl. That's the carbonyl of the fatty acid on each of those positions there. And then you've got this final methylene terminus, and that particular, which was once also OH group, because glycerol is 3 alcohol groups, right? But now you've got a phosphate ester there, and that's going to be whatever that head group is. So the X can be the following. It can be water, ethanolamine, which we saw recently, choline, the amino acid serine. It could be a sugar known as myo-inositol. That's what myo-inositol looks like. It's hexose sugar. It could be glycerol itself. So you can have phosphatidylglycerol, where you have a glycerol attached to the 3 position, but you still have the glycerol backbone, understand, right? And that's what we're talking about here with phosphatidylglycerol. That's what it looks like, you see, showing you. So the name of the lipid, if it's just water there, it's going to be phosphatidic acid. If it has ethanolamine, it's phosphatidylethanolamine, phosphatidylcholine, etc. Right? See, choline is something more than trimethylated ethanolamine. Those methyl groups, by the way, come from acetylmethionine through folic acid metabolism. Okay, so sulfur metabolism and folic acid metabolism link up to phospholipid biosynthesis. And we've talked about this in the past. Now, this diphosphatidylglycerol has a glycerol on both ends here, you say. This diphosphatidylglycerol is basically a 2-component lipid, and it's got a very special name. It's called cardiolipin, and cardiolipin is the major lipid you find in the inner mitochondrial membrane in animals. Okay. It's named, of course, because the first time it was isolated was from cardiac muscle. Okay. So there's a lot of diphosphatidylglycerol or cardiolipin in heart muscle, as you might guess. Now, talk about the membrane macromolecular structure now, okay? I'm zooming out now again. Way back in the 70s, Singer and Nicholson said that their idea of what the membrane looks like could be described as a fluid mosaic. A mosaic of what? Just like in art, a mosaic of various components. And those components are going to be unique lipid structures that will ornament the external leaflet or the internal leaflet, shown here, these so-called glycolipids, right? But you also get internal lipids into the cytosol. So that's part of the ornamentation. And this carbohydrate moiety, either in the extending from the inner leaflet into the cytosol or from the outer leaflet into the extracellular matrix or milieu, that carbohydrate moiety is going to be responsible for some of the binding of ligand to proteins. And the proteins, of course, are going to be what? They're either going to be glypiated, that means they're going to have glycolipids covalently bound to them, or they're simply going to be glycosylated polypeptides, carrying with it things like sialic acid, which we talked about before, right? Yeah, much carbohydrate. And various other kind of hexoses, ornamentation. We talked about glycolipid, glycoprotein biosynthesis happening in the endoplasmic reticulum at great length, several lectures. So I'm not going to belabor it right here. What else Singer and Nicholson said back in 1972 when they published this paper was that you got a bilayer membrane, you've got cholesterol interdigitated within that membrane. And because of kind of studies that were being done at the time, it appeared that when you add cholesterol to the membrane, it does a couple of things. It makes the membrane a little bit wider, okay, giving it actually more surface area in three-dimensional space, but also causes some ordering of the acyl chains when it is cis to those acyl chains. And that ordering will slow down the fluidity of those long chain aliphatic hydrocarbons. And that will allow for the resolution of water channels that are dictated essentially by that interaction between the cholesterol moiety interacting with the glycerol lipid or the sphingolipid, depending on what the composition of the membrane is. Okay. So that's their basic model there, right? The Singer-Nicholson model. So they also said in this paper that the lipid phase can be either liquid crystalline or gel, and it's typically represented as a bilayer hydrophobic fatty acyl core. That's what these blue hydrophobic chains are shown here, right, throughout the fine length of the membrane. It's a cross section, right? And then the polar head groups were considered to be on the surface because they interact with water. And indeed, that's what you find. That's what a bilayer would look like, essentially. And then you got the sterile lipids, are mostly cholesterol, really. You see that interdigitation. And you also see the membrane proteins. They threw those in as well. And they say these membrane proteins can be completely embedded in the membrane, or they can have surface ultrastructure outside into the extracellular matrix. And they can also have a component intracellular. So the protein can be large enough, fits in the membrane, but it can interact with the extracellular matrix as well as the intracellular cytoplasm. Okay? There are some proteins like that, obviously. Then you have all kinds of channels, sodium channels, potassium channels, etc., which we could talk about as we move on, right? So aqueous dispersed emulsions also generate a non-bilayer phase. So you have lipids and you disperse them in water, you will make a bilayer, but you'll make more than a bilayer. You'll make other kinds of structures. Sorry, dog. He'll stop soon. So this is an important point here. More solid gel phases occur at lower temperatures. So rather than having a fluid liquid crystalline type of membrane, which is considered more biologically dynamic because it has that fluidity, and fluidity is a rheological term which describes essentially the punctate transmissional alteration around that membrane. What I mean by that is that the membrane can be deformed. And if it can be deformed, you can change its microstructure. And if you change its microstructure, you change what? You change its function. Right? Another very key component here. So you're starting to get to some of the detail. But keep in mind, as you lower the temperature, unless you have, of course, a homothermic organism like a human where we generate our own heat, where we maintain a body temperature, you very seldom will get a membrane that will go into the gel phase, which is a more solid phase, mind you, okay? But if you're talking about poikilothermic organisms, like microorganisms that are living free, that is, not inside the gut, or you're talking about plants, green plants, which are poikilothermic, they don't generate their own heat, then obviously the temperature fluctuations that can occur even within a day are going to dictate a tremendous amount of physiological consequence to what that plant is doing. And this is exactly what occurs in terms of the regulation of photosynthesis and photorespiration, as well as growth and development of the plant. Okay. And sometime I'm just going to spend, I think, a whole week on lectures just talking about plant physiology and plant biochemistry, because that's how I started out as a plant biochemist. And then I moved into animal studies subsequent to that. Right? And so plant physiology and plant biochemistry is actually more complex than animal, because plants do a whole other function, because they are autotrophic. That is, in the case of green plants, they're photosynthetic. So you have more physiology, more interaction, and also more unusual structures for membranes. Right? Indeed. All right, so let's go through the rest of this. The longer the saturated alkane series, so the longer that you get, C6, C8, C10, C12, that's what it means, adding on the carbon units, the higher the melting point of the phase. Okay. Now, it was learned that there were problems with the initial model, and so it was immediately posed in the literature that since model lipid bilayers were organized in vitro using homogeneous glycerolipids like PC, it could be that when you have a native membrane from a cell, which is not going to be homogeneous with a particular lipid, just a polymer or a macromolecular structure of phosphatidylcholine, that you're going to generate all different kinds of microstructures. And indeed you do, right? Because there are no membranes in biological systems which are pure phosphatidylcholine, phosphatidylethanolamine, sphingomyelin, whatever it happens to be. There's mixtures. And because you have those molecular species of lipids, remember changing out the fatty acids in the 1 and 2 position of a simple glycerol molecule, or just putting what was in the 1 position in the 2 and the 2 in the 1, tremendously affects the morphology and therefore the structure and function of the membrane itself. It also alters freezing point, boiling point, and all kinds of rheological issues associated with, fluidity. Okay. This has been well studied. People have been studying lipid biochemistry for a very long time. In fact, many of the analytical tools we have in the biochemistry laboratory were developed to study lipids, such things as liquid chromatography, gas chromatography, mass spectroscopy, were really first dedicated to study lipid components, because it was very difficult to measure them any other way, because they weren't aqueous, so we couldn't dissolve them in aqueous. And organic solvents have this tremendous problem. They're notorious for absorbing UV light at low ranges. And UV light is often used to follow, because of the way that organic compounds absorb and transmit light, easy to follow such things as amino acids, carbohydrates, etc., during the purification, during a chromatographic separation. And you can't do that so well when you've got lipids, because lipids are already dissolved in organic solvent. Organic solvent absorbs a lot of UV light. So you just get a wall. You can't see peaks, you see. So then different tools had to be generated, such as mass spectroscopy, rather than a UV detector, an ultraviolet light detector. So that's, again, I learned that from working with lipid biochemists back in a USDA lab years ago, the Northern Regional Research Center. It's been renamed now, but it's in Peoria. And people working in that lab actually perfected a lot of the very early lipid work using mass spectroscopy. You go back and look at those papers starting in the 70s and moving all the way into the 90s and on. All right. So we had compositional macro models that equated perhaps with tertiary, and we know we have macromolecular structures, and we know we have this tertiary and quaternary structure of the membrane, right? So if you think about proteins, we talk about tertiary structures like the folding of a protein into a globular settee, or the interaction of two different globular domains making a quaternary structure of a protein. We know we have that with the macromolecular membrane, because that's what we observe. But we have yet to really get a primary structure of the membrane. And that would be what? Not the composition, but the sequence. And so because we don't have good sequence information to date, we're still not sure about the real biology, and also, I would say, a great deal of the biochemistry of the membrane lipid itself. Okay? That's a very important thing. I just want to show you the earlier model, the Davison-Danieli model, shown here is back in 1935, where they assumed that you had proteins stacked on the outside of a bilayer. That's what's supposed to be. These purple discs here apparently are supposed to be proteins. So that model was discarded. This model then came about. And this model still has a lot of significance because it still basically represents kind of what a membrane looks like. But we have much more detail now. We also know there are multiple different phases that lipids will take on, therefore generating different kinds of membranes. Okay. So with that, I want to now switch to you for you to understand that membranes are just like with proteins, aren't composed just of interdigitated specific lipids, which I said right now we still don't have a sequence for. Right? For example, we don't have a sequence for the plasma membrane of a cardiomyocyte. Okay, we don't have the sequence of the plasma membrane of an hepatocyte. But we do have sequences of, say, I don't know, choose your favorite enzyme, right? Catalase, peroxidase, glucose-6-phosphate dehydrogenase. We have complete sequences of those proteins, like what amino acid follows another from the N-terminus to the carboxy terminus. And then from there, you get a secondary structure, right? Like alpha helices and beta sheets and whatnot. The tertiary structure, when the protein's folded, and then that folding itself can be spontaneous typically. Sometimes chaperones play a role, but not very often. And then again, if you have two totally different peptide domains interacting one next to the other in association, sometimes covalently, that's when you get quaternary structure of a protein. So it's funny because in lipids, we can see the tertiary and quaternary structure, but we don't know the primary and secondary very well. Right? That's the point I'm making. So it's kind of funny, really, because lipids were some of the first things we tried to dissect and understand because they're so fascinating. But because they're fascinating, that means there's a reason for that. And the reason that they're fascinating is because they're very, very difficult to study because they are not soluble in aqueous. So you can't manipulate them like you can aqueous dissolved organic compounds, which are a major tribe of biochemical analyses. Okay. All right. So just like with proteins having domains, structural function domains, it turns out that the lipid membrane also has structural functional domains. And we call these sometimes membrane lipid rafts. Talked about those a great deal this last year, particularly involved in various kinds of pathophysiological diseases, right? So I'm not going to say anything else about that until we get back to it. Remember the membrane lipid raft. So you have raft domains, and they involve sterols, such as cholesterol, sphingolipids, which I haven't shown you a structure yet, but if you don't know, I will show you, don't worry. And these correspond to patches or regions or domains of lipids, these rafts, right? And they're in a liquid ordered phase, usually, a liquid crystalline state, not a gel, within a matrix in a liquid disordered phase. So whenever you have a membrane lipid raft migrating and fusing to a membrane, and we talked about this when we're talking about periodontitis in the audio lectures, you have bacterial membrane lipid fragments that fuse to, say, the epithelia of the gingiva in an oral cavity of a human. You, of course, are introducing this lipid raft, and then you're changing the morphology of the membrane. And remember what havoc was wreaked from that. Basically, alarm bells going off of G-protein coupled receptor responses, inositol phosphate cascades, corruptions of sodium, potassium, voltage gated channel transport, ultimately leading to stress responses, turning on, for example, the UPR, the unfolded protein response, therefore dysregulating the secretion of something like an immunoglobulin or a cytokine or a chemokine or a growth factor or a matrix metalloprotease. You see? So the same kind of thing can happen whenever an endogenous membrane lipid raft melts into a plasma membrane, this time coming from the cytoplasmic inner leaflet surface. Okay, so knowing that, I want you to understand that not just the plasma membrane can be added to and subtracted by lipid membrane rafts, but so can all the endogenous endomembranes. Particularly think about the nuclear envelope, what that might do to alterations in gene expression and the function and transport of RNA, including microRNA, regulating the gene expression in the cytoplasm because of the RNA-RNA hybridization that occurs with messenger RNA. Okay? So membrane lipid rafts have a huge story. We're developing it, but we are really in the very early stages of describing it. I would say in the next 10 to 20 years, we're going to know much, much more about lipid membrane rafts and what they do to cellular function, even at the level of gene expression, changes in bioenergetics, etc. Okay? So that's where I wanted to basically get you on that point. So if we look at some of these membrane structures, that's what these are here. You can have, this is a very basic cone-like structure. You can have micelles where the interior is completely hydrophobic and the exterior is hydrophilic. Then those micelles can generate, depending on the chain length of the fatty acid and the intensity of the electronegativity. So that is the cationic or the anionic properties. Cationic has a positive charge, anionic a negative charge on those so-called polar head groups. And the chain length can be modified by the global interaction of those polar head groups with each other and the acyl chains associated with it in regards to the number of double bonds and the level of cholesterol that is there. You can generate an aqueous center. So that's another kind of membrane structure. Okay? So you have a micellar structure which can transport bulk substances such as very hydrophobic substances, other lipids, right? Then you can also start now getting more and more differentiation of that aqueous center so it becomes essentially columnar and more like a channel, which will then interdigitate, intervaginate into the inner and outer leaflet of the membrane. And notice these membranes are all shown closed because that's what happens with lipids. They close upon themselves because they curve in association with the amphipathic nature of the phospholipids, which are the composition of the membrane lipid. So sometimes you make bilayers, sometimes you make various other structures. And we are going to start naming these other membrane structures right now. So depending on the kind of lipid you have, that's what we're showing you here. So this is monogalactosyldiacylglycerol. That's monogalactosyldiacylglycerol, monoglucnac diacylglycerol, phosphatidylethanolamin, serine, phosphatidic acid, and diphosphatidylglycerol or cardiolipin. All of those lipids will tend to make these cone-shaped structures, right, because of the interaction between the polar head group and the acyl chains. And when you get a massive amount of these lipids, you will make a hex 2 phase in cross-section. That's called the hex 2 phase because of its molecular geometry. You can also, depending on how much water is added to these structures, a so-called cubic phase, where you have this highly digitated intramembranous structure, largely encased around hydrophilic arranged core, right? If you have digalactosyldiacylglycerol or or the diglucosyl diacylglycerol, the sulfoquinavosyl diacylglycerol, phosphocholine, phosphoglycerol, phosphoethanolamine, you'll make your typical bilayer, okay? The bilayer, or the lamellar phase of the two membrane fractions separating out, with the interior being hydrophobic and the exterior being hydrophilic. Okay, those as a lamellar phase, right? And also, if you have a lysolipid, that means one of the fatty acids is missing in either the sphingosine base or the glycerol lipid base. You're going to generate something called the hex 1 phase, right? Which, instead of having this huge core here and then all the acyl chains spilled out, what you get here is much more tightly packed with an inner, much more defined and refined region of an aqueous compartment. So you have hex 2 phase, hex 1 phase, the classical lamellar Singer-Nicholson bilayer membrane, and you also have cubic structures. All of these can be found in even one membrane, such as the plasma membrane, hence generating different domains and therefore structures and therefore functions. And remember, this is not a fixed thing. It's not a static arrangement. These lipids are constantly turning over and rearranging, and with the help of the mitochondria and the endoplasmic reticulum and even the peroxisome, and the association with lipid membrane raft transport in and out of the plasma membrane, you have a constant alteration of all these different structures in the membrane in some kind of topodynamic existential event ontology that we have not yet measured in real time, because we don't have a primary structure of the membrane. So these are what we see like after the fact. After we synthesize these purified membranes, we can discern these structures when we alter things like sodium concentration, potassium concentration, and water concentration. But we have no idea how the actual sequence of events occurs, because we don't have the actual sequence of the macromolecular membrane structure, like what lipid follows the other in some kind of plane, right? Longitudinal plane, right? So either horizontally or vertically, three-dimensionally now, right? And also in a temporal framework, because these lipids turn over all the time. So all 4 dimensions are functioning, all the spatial dimensions and time. Okay? And very rapidly, membranes turn over, particularly if they have a lot of polyunsaturated fatty acids. So shape structure concept of lipid polymorphism is what I'm showing you here. Lipids with small polar head groups have a molecular shape that resembles a truncated cone. They induce a negative curvature strain on whatever membrane they're going to make, and they favor the organization of membranes into inverted, they're inverted because the acyl chains are out rather than in, micellar structures, or cubic bicontinuous structures that I already described to you. The lipids with a bulky polar head group and only one acyl chain have a molecular shape similar to an inverted cone. It's down here. It's the lysolipid, right? And it induces a positive curvature. So you can have a negative curvature or you can have a positive curvature, depending on the acyl chains interacting with each other, the length of those acyl chains, in productive ionic hydrophobic turnover events with the polar head group and the aqueous milieu, and of course all the proteins embedded within, which can be either all the way from hydrophobic to hydrophilic, and every protein is like that, various regions, depending on the amino acid sequence, right? So they favor the formation of this tubular H1. That's what this is here. I already described that to you. Or basically spherical micelles. It's more like a stacking of micelles, like the typical micelle I showed you in the last slide. Right? And again, when you have your normal sort of polar head group, you have both fatty acids, you make that lamellar phase. Bilayers. Bilayers is still very significant. We're not discounting it in any way. Okay. This slide shows you now some of the lipid composition of various membranes. The human erythrocyte versus myelin versus beef heart mitochondria, and then a bacteria, gram-negative bacteria known as Escherichia coli. So there's basically no phosphatidic acid in beef heart mitochondrial membrane or in E. coli, but the human erythrocyte has about 1.5 mole fraction, where human myelin sheath has about a half. Now look at the disparity there relative to, say, sphingomyelin. Sphingomyelin, about 17.5 molar equivalent in the erythrocyte, only 8.5 in the so-called described axonal myelinated sheaths in neural bundles, right? So you actually have more sphingomyelin in the erythrocyte than you do in myelinated neurons, right? But that's where they were first described. Then you can look at different glycolipid concentrations, that is molar equivalents, I should say, and different cholesterol concentration. Of course, there's no cholesterol because prokaryotes don't make that sterile lipid, presumably to do with the amount of oxygen in the environment when they evolved, according to one of my mentors, Conrad Bloch, who's now passed away a long time ago. You also see very low levels of cholesterol in the mitochondrial membrane. In fact, the cholesterol content of membranes increases as you go from the internal part of the cell, if there is such a thing as internal, that's where most of the organelles are centered, and you move out through, say, the nuclear envelope to the mitochondria, to the endoplasmic reticulum, to the Golgi apparatus of the plasma membrane. If that is indeed a vectorial telos from internal to external, and it can be described as that from cross-sectional analysis, the cholesterol concentration increases as you go out from there. Okay? So you have about 50 mole percent cholesterol in the plasma membrane, and you only have like 10 or 15% of those intermembranous systems. You understand? That has a tremendous effect on function. Plus, also allows the cholesterol to be in the plasma membrane where it can be metabolized to generate glucocorticoids, mineralocorticoids, and sex hormones. Right? All of those really interesting steroids you've heard probably a lot about. Many of you, I'm sure, know what I mean by the structures of those 4 different classes. Now, I want to show you here that at the same time you've got these individual lipids, you have to understand this is biochemistry, so we're going to talk pathways, right? There are nested biochemical pathways that are found in the endoplasmic reticulum, the Golgi apparatus, even to some extent the mitochondrial membranes, and even some in the matrix, which catalyze the biosynthesis of these individual lipids. And of course, you're going to have crosstalk between lipids, and this can occur in the membrane, but also in endomembranous compartments and organelles, like I just mentioned, mitochondria, endoplasmic reticulum, Golgi apparatus, peroxisome. So one such important reaction for phosphatidylcholine biosynthesis is this Cytodeal Transferase. So I want to talk about this enzyme here just for a moment. Take a look at what's going on. So choline, which is this trimethylated ethanolamine, can be transported by different kinds of proteins that is in circulation, okay, but also transmigrating across tissues and into cells. Once choline is introduced, there's an enzyme called choline, but also it will react with ethanolamine. Remember, choline is just trimethylated ethanolamine, right, which comes from serine, right, after decarboxylation of that amino acid, don't you know? So these kinases then will generate choline phosphate. Choline phosphate then can react with ATP, UTP, and the amino acid glutamine to generate ADP, PI, and glutamate. And now you've made this transfer of CTP, and you've removed pyrophosphate. Now you've made a nucleotide polar head group. This is CDP choline. So the product of this choline phosphate cytodeal transferase is CDP choline. It's a nucleotide, right? That's cytosine diphosphate choline. Right? The only thing you need now is diacylglycerol, which can come from lipase-mediated removal of one of the fatty acids, the 3 position exactly of a preformed glycerol lipid from triacylglycerol, and that enzyme is called CDP choline diacylglycerol choline phosphotransferase. Right? And now you've made PC. This is one way of making it. There are other ways, right? All right. So Cytodeal transferase is actually upregulated by DAG. So for this enzyme to be fully functional, you have to have sufficient amount of diacylglycerol. There's no polar head group on that. This is a feed-forward reaction because it's also a substrate. You understand. That lipid activates that enzyme and then utilizes that lipid, right, to drive the CDP moiety away and make phosphatidylcholine. You've added the entire diacylglycerol by removing the nucleotide at the diphosphoryl level, right? And that's how you make PC. So CT is upregulated by DAG, and that directs the enzyme. What the DAG actually does for this enzyme is it loads it to the endomembrane, okay? It directs the enzyme to the endomembrane. So because CT, cytodeal transferase, that reaction is only active when it's in the membrane, okay? When it's within the membrane. So this now brings on another really cool lectural nugget, and that is you have this amphibolic nature of metabolism. What do I mean by that? That means that you have preformed molecular pathways. For example, this pathway making phosphatidylcholine. And the enzymes are there, and the pathway is there in terms of its transcription translation. But it's not functional until you move those enzymes to some subcellular compartment. So there are many enzymes like this. They either require to be removed from a membrane to be active, say, in the aqueous milieu, or they're required to be moved to a membrane so they react where the substrates are. So there's a lot of fractionation of metabolic pathways associated with lipid metabolism, sensu stricto, like biosynthesis, but also lipid membrane raft migration. So it is a tremendously dynamic event. There's nothing static about this system at all, right? That's why lipids are so interesting, as I like to say, because they're really driving overall cellular structure and function and even fate transition, right? At a level that was basically opaque to us until we started studying these enzymes, using detergents like Tritinex 100 to isolate an enzyme like the cytodeal transferase. You see? It's the kind of thing that us lipid biochemists do in the laboratory. Coax membrane bound enzymes out of the membrane, then we have to immediately put it in the right kind of solution so it remains active, sometimes by using things like acetone in the buffer, so that the enzyme thinks it's still in a lipid milieu, so it will be still functional, so we can study it. Right? Look at its enzyme kinetics, look at the turnover number, look at the Km and Vmax, all that, what you do when you study enzymes. So that's why lipid biochemistry is also very complicated to do. Not just complex because of the biology, it's complicated to do lipid biochemistry because you're constantly moving from organic solvent to aqueous solvent systems, because many of the substrates and products can be aqueous dissolved, not lipid dissolved, but all the intermediates are not. Or sometimes you go from complete aqueous to complete hydrophobic. And you have to make that work all during the study of the enzyme. Right? Of course, you can do radio labeling and you can do it within cells, but what kind of cells? And that limits what you can really do, right? I just wanted to bring all that out. Now, diacylglycerol would accumulate during hormone-sensitive lipase activity, which actually drives the synthesis of free fatty acid, which can be reesterified to the thioester coenzyme A for such things as complex lipid synthesis. For example, the synthesis of all the phosphoglycerolipids and sphingolipids, and also the alkyl lipids, right? The ether link lipids. But the other thing it does is allow that fatty acid to be used for bioenergetics, because a CoA thioester of a fatty acid can be translocated into that mitochondria via the carnitine palmitoyl transferases, and from those series of reactions lead to beta oxidation of fatty acids, and of course the production of NADH and FADH2, which can be reoxidized in the electron transport chain via the proton motive force across the intermitochondrial membrane, ultimately pushing through the F0 F1 ATPase of complex 5, last component of the oxidative phosphorylation electron transport chain driven mechanism for ATP synthesis de novo. Right? So you understand, these are all linked. Bioenergetics are linked to the production of the membrane lipid. And the question is, where's the regulation, right? Well, I just told you part of it. Diacylglycerol mobilizing that cytodeal transferase into the membrane where it's functional is linking coherently that lipolysis reaction, yielding a specific membrane lipid, which will allow reorganization of a membrane, endomembrane or plasma membrane, in such a way that you can modify gene expression because of the carrying of, for example, transcription factors into the nucleus, and then chromatin retailoring and epigenetic modifications sometimes leading to the production of specific transcripts and then, of course, translation back in the cytosol and the endoplasmic reticulum to make polypeptides that drive a given cellular response, such as an immune response. Okay. You see how all this comes together now, I'm sure. You're starting to see why it's really a cool thing and why it's really fascinating when you start tearing it apart like this, because this is the whole thing. This is really what you do when you start looking at authentic biochemistry. It's not a simple thing where here's this pathway and here's its regulation. You need to know all of that because it is authentic. And as much as we've been able to reproduce it, it appears true to the nature of the science we're doing. But then you have to add in every other component that's occurring simultaneously or sequentially or prodromally to be able to have not a static cellular tissue organismal event, but a completely dynamic one that's living until it dies. So that's this whole process what I'm trying to say. So all these things I'm showing you look like static phenomena, but understand they're constantly changing and moving along, but not in a stochastic way, not in a random way, but in a very ordered way, a providential way, if you want to put it that way. Right? And that is related to the genome, the organization of the genome, and what the genome is comprised of in terms of the genes, and the exons and the introns and the shuffling of domains during evolutionary processes to cause speciation in different organisms so that they then end up having different kinds of patterns to generate differentiation and development so that you get a tadpole versus a spermatozoa, for example, right? But the biochemistry is at the foundation of it, right? It's the architectonic of the whole process. That's why I really try to emphasize when I can take it all the way up to that level of talking about cellular interactions and then deeply way down discussing individual biochemical modifications leading to such things as changes in gene expression. So I'm going to stop there. I think I've done enough time, hopefully not boring you today, with this little membrane lipid lecture. I've got 2 or 3 more I'm going to do. I think I might intersperse these with more audio authentic biochemistry because I want to move forward in that process I'm doing there. But I think I can complement it by continuing on with this lipid membrane. And you can see why I'm doing this in video, because doing this kind of thing in audio and not showing you these interactions, I think would be a terrible malfeasance on my part, that you don't understand it because you're not seeing these structures. You need to understand the structures to understand the function. And if you take biochemistry from me, we emphasize both those things. We emphasize the structural components and the functional components, thus generating that dialectical event ontology, which is what I think biochemistry really cognately displays for biological systems. All right, so let's get out of this show here and come back to our box where we can shut things off. Let me just say that this was Dr. Dan.J.Guerra from Authentic Biochemistry Studios, finishing this lecture on the 21st of January 2022. And I will be back to see you soon, both in this video format, and you'll also hear me on the audio version of Authentic Biochemistry. Please subscribe both to the YouTube channel where you're watching this and also to the podcast, which is on all the regular platforms. And so I'm sure you'll be able to find it. I need more subscribers. I need more support to be able to do this. I'd really like to get more people watching this so that I can expand this and really get into doing a public series of lectures, which basically are free, but I still need some kind of support to be able to spend the time to do this, as you might understand. And then we can get also something set up where we'll do interactions, right? I certainly already will take email, and you can associate comments either on the podcast web pages or on the YouTube web pages. I read the comments and I try to respond to them if I can. All right. So Friday night, go out and have some fun, right? Dr. Dan.J.Guerra saying bye for now.